Our next speaker is Bob Weinberg. Bob is American Cancer Society Research Professor and Daniel Ludwig Professor for Cancer Research at MIT and Director of MIT's Ludwig Center for Cancer Research. In 1979, Dr. Weinberg and his colleagues provided the first demonstration that the genomes of chemically transformed cells carry a mutant oncogene, which they subsequently demonstrated it was a RAS oncogene. And I remember vividly reading that paper. I think it was in PNAS, where they so elegantly demonstrated that you could just extract DNA from cancer cells and by calcium phosphate precipitation add it to more or less normal cells and confer that transformed phenotype to the normal cells. And it was an amazing publication. Several years later, Bob showed that this oncogene differed from a corresponding normal gene by a single nucleotide change in specifying the 12th amino acid of the RAS P21 protein, thereby discovering for the first time a mutation that is causally important in human cancer formation. In 1986, his laboratory participated in the isolation of the first known tumor suppressor gene, the retinoblastoma gene termed RB. In 1999, his laboratory created the first genetically defined human tumor cells utilizing the recently cloned gene that encoded the human telomerase enzyme. And in 2004, his lab demonstrated that Twist, a gene normally involved in early embryogenesis, is used by cancer cells in order to acquire invasive and metastatic traits. His current work is focused on the mechanisms by which cancer cells develop highly malignant traits, specifically those that underlie the complex processes of metastasis. I've had the pleasure of introducing Bob on several occasions, and even though he asked me to keep this really short, I always like to highlight one publication that I think doesn't necessarily get quite the recognition it deserves, but was extremely important for a lot of women who have been treated and gone on to survive breast cancer. Around 1985 or 86, Bob, in collaboration with Michael Green, published an incredibly important paper that showed that if you developed an antibody to the HER2 gene product, and this is a transmembrane protein that can drive oncogenesis and does drive oncogenesis in about a quarter of women with breast cancer, HER2 overexpressed breast cancer, for which Genentech has developed a drug, Herceptin. But their demonstration was that an antibody could stop or dramatically slow the growth of such malignantly transformed cells in rodents. And that provided really a very strong impetus to encourage us at Genentech to move that project into development. At the time, there were a lot of competing projects that all looked kind of good in theory, but until Bob's present publication that this antibody actually can just shut down these transformed tumors in rodents, I'm not at all sure that project would have gone forward. So on behalf of many, many people, I'll thank you for that publication, and it's a seminal one. Bob is a member of the U.S. National Academy of Sciences, the U.S. Institute of Medicine, and a fellow of the American Academy of Arts and Sciences. And among his many honors, and he's going to shoot me for this, but are the Discover Magazine 1982 Scientist of the Year. I'm almost done. The U.S. National Academy of Sciences. And here's Bob.
Well, Art, if I had the presence of mind, if I had the presence of mind in 1985 to patent the work on the HER2 new oncoprotein, I'd be wearing really nice clothes right now. Thank you all for coming. I was told that this would be largely a lay audience, which of course is highly inaccurate, and to prepare a talk which, in fact, would be intelligible to the intelligent layman. And that's what I did to talk about the history of this field and indirectly my involvement in it over the last decades. And perhaps this will provide a useful foundation to the other talks that we'll hear this morning and this afternoon. I say that all with apologies to those who are already deeply involved in ongoing cancer research, because much of what I'm going to say really reaches back a number of decades, and I kind of had the illusion, which I still have, that you would be interested in to know how we got to where we are, how we are here now, almost 4 decades after this field began. For me, the really important input into my thinking in the mid 1970s was the realization that a whole series of different known chemical agents, we list them, were etiologically responsible for the formation of human cancers. And this really was a major puzzle because one really had no idea at the time how chemicals could actually induce cancer. The slide, where's the slide advance? How do you advance this? Oh, maybe this one works. At the time, one knew already, indeed, from the 1930s, that a whole series of chemicals could induce cancer. These are cyclic hydrocarbons. They were known already from the 19th century to be highly carcinogenic, but their mechanisms of action were really totally obscure. How could a chemical actually induce the formation of a human cancer? For me, a great conceptual inspiration was the work of Bruce Ames, then and now at UC Berkeley, who demonstrated in 1975 that if one looked at a diverse array of different human carcinogens, that their ability to induce cancer, indicated here on this logarithmic plot, could be correlated over 4 or 5 orders of magnitude with their ability to induce mutations. And that led in turn to the insight that carcinogens cause cancer. It was still a speculation, through their ability to actually damage genes. At the time, obviously, that was only a speculation. And the further conclusion, deduction, that cancer cells must in one way or another contain mutant genes that were responsible for the aberrant behavior of those cells. Again, this was simplistic reasoning, but just because it was a model, as I often say, doesn't mean it was necessarily wrong. And so just to recapitulate some of and elaborate on what you just heard, starting in 1978-79, my laboratory began to investigate the nature of chemically transformed cells, which putatively had in them the mutant genes that were responsible for the aberrant behavior of these cells. At the time, we knew of the work of Varmus and Bishop, here, obviously, at UCSF, which indicated that retroviruses could activate certain cellular genes, but it was already clear at the time, from very intensive research in the mid-1970s, that retroviruses were not etiologic agents in the great majority of commonly occurring human cancers. And so at the time, we undertook, because we've been working on gene transfer, transfection since the early 1970s, to take DNA out of chemically transformed cells, prepare the DNA, and introduce it into normal mouse cells with the hope that the normal mouse cells would respond to the introduced DNA and thus genes by undergoing the process of malignant transformation, which one could actually observe in the Petri dish as a focus of cells which grew morphologically in an aberrant fashion, and which one could see here, this is what they actually looked like, these foci of transformed cells, and which, when implanted into a mouse, actually could form a tumor. Such tumorigenic ability in vivo rather than in vitro was the ultimate validation of the fact that the cells had been converted from a normal to a neoplastic state. And it began to provide solid evidence that indeed the genomes of chemically transformed cells and putatively human cancer cells were actually carrying mutant genes that were etiologic, that were causally responsible for the formation of neoplastically transformed cells. And so we began to develop, we and others, several other groups began to work in this area as well at the time, to try to figure out the basis, the molecular basis of this transforming principle. In 1982, we and a second group, that of Michael Wigler, cloned the gene that was responsible in a human bladder carcinoma for the transformed phenotype of those bladder carcinoma cells. The carcinoma cells had been extracted from a 55-year-old man who had been smoking since he was 15 years of age. And ostensibly, the chemicals from the cigarette smoke were metabolized, introduced via the urine into cells in the wall of the bladder, where they apparently introduced some kind of mutation. At the time, we therefore cloned the corresponding normal proto-oncogene, using the Varmus and Bishop nomenclature here, and we found that the two genes, that is the gene isolated from human bladder carcinoma, here in the red bar, and the one from a corresponding normal human DNA, were essentially identical, indistinguishable, yet, as one can see here from these results, only the cloned oncogene carried the information for inducing the transformation of cells. And pursuing that, we and a second group that of Mariano Barbasid, reported the fact that if one looked at the 6,000 base long clone gene, there was in fact a single mutation that was responsible for converting the normal proto-oncogene into an oncogene. And so there was a very subtle difference already in these pre-massive cloning days that one could ascribe to the two genes, one critical point mutation out of the apparent 3 billion bases of DNA that one imagined lay there at the time. And this represented an enormous conceptual simplification for us, but obviously was not on its own the answer to cancer. We ultimately learned that this controlled a oncoprotein whose continued activity was potentiated by this mutation and the resulting substitution, as you can see here, of a glycine residue with a valine residue. Later, actually, Frank McCormick, who was here, began to describe at a biochemical level how and why this point mutation and amino acid substitution actually led to the transformed state. And in the years that came thereafter, one discovered that in fact such point mutated genes, this oncogene, it turned out, was homologous to a retrovirus associated oncogene, and therefore, because it derived from rat sarcoma virus, was called RAS. Such point mutated sarcoma, Harvey RAS, KRAS, or NRAS genes could be found, as you can see here, in a diverse array of human tumors, but hardly all of them. And this began to suggest that this RAS gene represented a small part of the genetic aberrations that one could ascribe to human cancer cells, at least as viewed through this prism of mutations in the protein encoding sequence. Having said that, we discovered in 1983 what was perhaps for some an inconvenient truth, because the moment that point mutation was described, many people said that this cannot be the answer to cancer, because we know that tumorigenesis is a multi-step process, and therefore it was inconceivable to them, and indeed realistically so, that a single point mutation could on its own convert a normal human cell into a cancer cell. It obviously was more complex. And so in 1983, the work of Hartmut Land and Luis Barada showed that if we took truly normal cells, in this case recently explanted rat sarcoma cells, and put a RAS oncogene in them, nothing happened. The cells that had previously been studied had already been pre-adapted to culture and were immortalized, and therefore were not truly normal. If we put at the time a cloned MYC oncogene or an adenovirus E1A oncogene into cells, also nothing happened. But if the two were concomitantly introduced into cells, then this oncogene collaboration resulted in the formation of virulently growing cancer cells. And that suggested the principle that multiple genetic mutations must coexist within the genomes of human cancer cells that then collaborate one with the other to create the ultimate transformed phenotype, and that a single mutation on its own, which obviously we take for granted now, could not succeed on its own in inducing neoplastic transformation, at least in the mouse cells. Here, in fact, was the validation of that, because virtually all human cancers were known to have passed through a whole series of intermediary histopathological states. You can see those on the top, and take a very long time to occur. And this was totally incompatible with the notion that a mutation in, for example, a point mutation in the RAS proto-oncogene would suffice to create a human tumor. Indeed, as we found later, actually one required far more changes. In 1999, Bill Hahn was able to demonstrate that if one looked at how many changes were required to transform a human cell from normalcy to malignancy, it actually required changes in 5 distinct pathways. Rodent cells, by the way, only require 2 pathways. And that begins to suggest an intrinsic limitation in mouse modeling of human cancers, because it seems very clear to us that mouse cells, at least, and human cells, at least vis-a-vis the process of cancer pathogenesis, are wired up very differently. Here, these rat cells, 2 mutations or 2 introduced genes sufficed, but subsequently, in our hands, we found that at least 5 are required, as mentioned, to do the same thing in human cancer cells. In 1986, through more good luck rather than intelligence or hard work, I became involved indirectly in the isolation of the retinoblastoma gene. Steve Friend came into my laboratory, told me that he wanted to isolate the RB gene, and I was patient with him, but knew it was a quixotic fool's errand, because he knew nothing about cloning, and in any case, no one had any idea where the gene lay in chromosome 13. But sure enough, within less than a year, he and Thaddeus Drija of the Mass Eye and Ear Infirmary discovered the gene that was responsible for this very early pediatric tumor, retinoblastoma, and which represented from the work of Alfred Knudsen the paradigm for tumor suppressor genes. And so one could then put all this together to begin to rationalize what is known often in the field as the Vogelgram, which Bert Vogelstein, whom we've just heard, put together in 1989, in which he made the fundamental observation that the histopathological progression of a cancer was accompanied by, and indeed ostensibly driven by, the accumulation of a whole series of genes. Here's the RAS gene I told you about before, and here are 3 tumor suppressor genes which seem to be inactivated during the pathogenesis of many human colorectal carcinomas. And this, of course, represented strong validation that the formation of a human cancer, as Bert just laid out in great detail, was indeed at a genetic level extremely complex. It was not simply a single mutation or even a small set of mutations that was required. And indeed, one can now say, after the fact, that if cancer formation could be triggered by a single mutation or a pair of somatic mutations in one of our tissues, that we would all be covered with cancers by the time we were 3 years old. The complexity of this here derives directly from the fact that our cells are hardwired to resist neoplastic transformation, and therefore each of these changes represents breaching yet another barrier to the process of neoplastic transformation. At a biological level, we'll hear later from Hans Clevers, because he indicated quite dramatically in his biological work why this particular tumor suppressor gene was lost early in colorectal carcinoma pathogenesis. And that in turn begins to indicate that we understand at a cell biological level now how many of these various genetic alterations actually facilitate and collaborate one with the other to create the complex cell biological phenotype of actual human cancer pathogenesis. However, as of late, that is over the last decade, my laboratory has shifted its focus because we had the presumption, and it was no more than that on the basis of much of the work that Bert has told you and on the basis of the work of a large field of cancer researchers, we had an idea, at least an outline, of how a primary tumor was formed. That is, one could begin to lay out a whole series of signal transduction cascades, a series of mutant alleles, which collaborated one with the other, to create the primary tumor. But at the time, and it's not so different today, we understood virtually nothing about the last step here, that is the one of invasion metastasis. Indeed, there's the question that was outstanding then and remains so. If you look from the 1989 time to today, what happens here? Actually, how does a primary tumor proceed to seed metastases in distant tissues throughout the body? And indeed, does this require additional mutations beyond those that were required to form the primary tumor? That remains to this day an unanswered question. And so we leapt into this area with both feet on the basis of work of Jing Yang, who began to study what particular genes were expressed in highly metastatic carcinoma cells, but not expressed in corresponding non-metastatic cells, in this case, mouse breast cancer cells. And here she stumbled across a series of findings that derived from developmental biology, developmental genetics. In the 1980s and 1990s, and even in the first decade of this millennium, the developmental biologists, such as the Drosophila geneticists, said to the NCI, give us your money because what we're doing is really going to be useful for cancer research. And people like myself scoffed at this because we were sure that these people, being as they were poor as church mice, simply wanted to poach on the cancer funds in order to continue their work, which we were fully convinced was totally irrelevant to human cancer pathogenesis and our understanding of the mechanisms by which metastatic dissemination occurs. And of course, and obviously, we were wrong. They discovered a whole series of transcription factors, master regulatories of cell fate, whose expression was already apparent early in metazoan development. These different transcription factors I'll show you the cell biological program that they choreographed, were already invented 600 million years ago and have been implanted and conserved in all of the derived metazoan phyla in the subsequent years. And these various EMT-inducing transcription factors, which I'll describe in a moment, actually were responsible for a very interesting cell biological process. Because if one looked at, in this case, for example, a human transformed breast cancer cells implanted, in this case, in a mouse, one found that the breast cancer cells, being of carcinomatous origin, were therefore epithelial to begin with and expressed the protein cytokeratin. However, if one looked at the outer edges of these implanted cancers, out here, one discovered that these carcinoma cells, or their derivatives, lost the epithelial marker, cytokeratin, and instead now expressed a mesenchymal marker called vimentin. And this suggested the notion, it became more than a notion, that when cancer cells grow inside a patient, or inside a mouse in this case, some of these cancer cells may undergo a profound shift in their phenotype, moving from an epithelial state with cytokeratins, shutting that down and expressing instead of the cytokeratin vimentin, which is a mesenchymal marker. And this program was called the epithelial mesenchymal transition. And one began to see that the mesenchymal cells that derived via the ostensible transdifferentiation of epithelial into mesenchymal cells, these cells began to have a very aggressive cell biological program. Here you can see this green invasive mesenchymal carcinoma cell beginning to invade into the recruited mouse host stroma, suggesting that this was indeed actually important etiologically in the formation of invasive growth. In fact, the EMT program, the epithelial mesenchymal transition program, had been described earlier by Betty Hay, later chairman of cell biology at Harvard Medical School, or as they call themselves in Boston, the medical school, much to the distress of the other medical schools, but we won't go there. Anyhow, she discovered this. She had been studying this for many, many years, and I was often given pushback by developmental geneticists who said I shouldn't really be working on the EMT because really I'm a cancer biologist and this was really their domain and I had no right to trespass there. And I reminded them in recent years that Betty Hay went to Smith College in Western Massachusetts, where she was inveigled to become a biologist by my sister-in-law's father. So I have a hereditary right to work on this protein. Anyhow, I digress, I fear. The EMT program, as I implied before, here we see an early sea urchin embryo, is very critical for various stages of embryogenesis. Here we see this ingression or gastrulation in which epithelial cells become mesenchymal and become, in this case, the unlagen, for example, of the mesoderm. And in the case, and obviously the implication of all this is that this cell biological program, which is involved in early embryonic morphogenesis, and which is involved also in wound healing in the adults, becomes co-opted by carcinoma cells, which exploit it by reawakening this program in order to acquire, in one fell swoop, the multiple attributes that one associates with the EMT program, this being acquisition of motility and invasiveness, and a full shift in their overall cell biological attributes. And therefore, this raised in turn the following kind of question with which we're still wrestling. If we look at this last stage of what's called the carcinoma to invasive progression here, one sees that actually this subsumes a much more complex series of sub-steps in this process. It's often called the invasion metastasis cascade and involves the invasion of primary carcinoma cells into vessels, their exit from the vessels later on, their formation of micrometastases, and ultimately their formation of macroscopic metastases. And confronting this complexity, one could pose the following question. Given the known ability of primary carcinoma cells to activate their previously latent EMT program, how many of the steps of the invasion metastasis cascade indicated here could conceivably be achieved by a carcinoma cell that turns on this program? Or is it the case that before carcinoma cells leave the primary tumor, they need to acquire additional mutations beyond those that were needed initially for the formation of the primary tumor? And we're beginning to get some insight into this. In other words, the question of whether additional mutations are required in order to impel or propel carcinoma cells to move from the primary tumor site to the site where they form a distant metastasis. For the sake of discussion today, I'd like actually to divide this multi-step progression into actually two large steps. The first step being how cancer cells physically disseminate to form micrometastases in distant tissues, and the second step, which involves the outgrowth of the micrometastases into macroscopic metastases. I often mention the fact that between 30 and 35% of women who present with breast cancer in the clinic have hundreds to thousands of micrometastases in their bone marrow. 35%. But only half of those women, less than half, will ever develop metastatic disease. And that testifies to the fact that only a minute proportion of the initially seeded micrometastases succeed in growing out into macroscopic life-threatening metastases. And it suggests the possibility that the rate-limiting step in this process is not the physical dissemination of cells from the primary tumor, which may occur quite efficiently actually, but the subsequent ability of a cell originating in one tissue to adapt to a foreign tissue microenvironment of another tissue, such as a breast cancer cell trying to make a living in the brain. So here's one of a series of experiments that were done in my laboratory by Zuzana Keshkeshova and Yasmin Dekok, in which they took human breast cancer cells and they implanted them as a xenograft in a mouse. And as you can see here from the top left, these MCF7-RAS breast cancer cells, if you see the white dashed line, are actually quite benign. They don't metastasize, and they have a rather smooth boundary at the edge of the tumor. If, on the other hand, one allows these cancer cells to form a small tumor, one turns on some transcription factors for a couple weeks that induce the EMT into these cells, one shuts off those transcription factors, and one looks at the mice 10 weeks later, then one sees that the tumors arising 10 weeks later are highly invasive. You see how they've invaded around the adjacent stroma, and perhaps of greater interest to our discussion today, these cancer cells, which otherwise would not have formed significant numbers of micrometastases, small numbers of cells, or macroscopic metastases, if they're induced, for example, by this pair of transcription factors to activate the EMT program, after which time the transcription factors are shut off. And then 10 weeks later, if one looks at the mice, the number of micrometastases has gone from 1 or 2 up to 400, and the number of macroscopic metastases has gone from 0 up to about 100. And to me, this is the beginnings of a proof that, in fact, it may be the case that by turning on the EMT program in a primary carcinoma cell, one can propel cells from here all the way through the multiple steps of the invasion metastasis cascade. I believe that the EMT program can explain these multiple steps, but for me the major conceptual challenge still is, how do carcinoma cells that are seeded into a distant tissue, how do they make a living there? How do they figure out, how do they cobble together an adaptive program to grow from a micro to macroscopic metastasis? At the same time, the work on the EMT pulled us, in the form of Sender Raimani and Maiting Liao, into another area. Because here's the work of Michael Clark and Mohammed Al-Hajj, 2004, where they took a population of human breast cancer cells and they separated them by fluorescence activated cell sorting. Precisely how they did that is not so important. But these cells all originate, individual dots, from the same population of human breast cancer cells. But if one separates them, as they did here, one discovered that 200 of these cells suffices upon implantation into a mouse host to give a new tumor, whereas 20,000 of these cells fail to do so. And that already proved inescapably that within a population of cancer cells, cells can, even though they may be genetically identical, cells can reside in more than a single phenotypic state. Here they reside in at least 2 states with vastly different powers to seed the formation of new tumors. And here was the adaptation of this work in my own laboratory. Here I mentioned the fact that these cells reside in this FACS plot in the population of non-stem cells, because those cells of these tumor-initiating cells came to be called cancer stem cells. I use that term unashamedly. Here's a population of immortalized human mammary epithelial cells. Here's the majority population of non-stem cells, the minority population of stem-like cells. Indeed, they can perpetuate themselves in culture. And what Mai-Jing Liao and Sender Raimani undertook to do was to ask the question, for reasons which totally elude me, what happens if you take the non-stem cells here in this analysis and you turn on one of these EMT-inducing transcription factors? Will that have any effect on their stem versus non-stem phenotype? Now, mind you, when they first talked about this to me, I was very skeptical, if not highly reactive, because here we have two things that were being worked on in my laboratory: cancer stem cells on the one hand, the EMT on the other. And when a postdoc comes to you and says these two things are really connected, more often than not, you suspect this is somehow the hallucinations of an overly ambitious postdoc, rather than being solid science. But here was the evidence, and it was solid science. If you turn on an EMT program through the expression of either of these two EMT-inducing embryonic transcription factors, then these cobblestone epithelial cells undergo a morphological transformation to a mesenchymal state. But more importantly, as you can see in these two graphs here, the activation of the EMT program induces a massive shift from a non-stem cell into a stem cell state. And so this suggested something which we were able to solidify later, and that is that in human cancer cells, the tumor-initiating cells have high expression of EMT-inducing transcription factors, and even in the normal breast, there are versions of the EMT program that are active. For example, here are the stem cells that exist in the mammary duct of a normal mouse, and the pink cells are expressing the EMT-inducing transcription factor called SLUG by overexpressing it by a factor of 36. And therefore, the EMT program is actually involved not only in normal mammary epithelial biology, but also in the derived carcinoma cells, i.e., when carcinomas form, they appropriate the EMT program, the stem cell program, which operates in the normal mammary gland, as well, by the way, in humans, to organize themselves within complex tissues. Here's yet another evidence of that. Here's a whole population of normal mouse mammary epithelial cells. 10,000 are implanted into a mouse mammary stromal fat pad. Nothing happens. But if one takes these cells, exposes them transiently to an EMT program, and then implants them in a mouse mammary stromal fat pad, now there's an explosion of morphogenesis, indicating that one has indeed induced these cells to pass from a normal state into a stem cell state that empowers them to actually create an entire adult duct, indicating here at least 100-fold increase in the degree of their stemness. And so we are now at a stage where we can begin to understand this complex interplay between what happens in the stem cell program in a normal tissue and, for example, in a breast cancer, where it becomes increasingly apparent that there's a slight variation on the EMT program as cancer cells pass from a normal mammary epithelial state into a breast carcinomatous state, but the same basic program operates. The program is intimately involved in the physical dissemination of cancer cells from the primary tumor. But what remains the major hurdle for us for now and the future is the question I raised before. I think we can now begin to get some feeling for how carcinoma cells physically disseminate, but we still have a very poor idea how they make a living in various distant tissues in which they seed themselves. And that, I think, will be a problem which will take years and years to figure out, because it could be phenomenologically very complex. Thank you for your attention and for coming today. Yeah, so I actually finished 20 seconds earlier than Bert did. I don't want to be competitive or anything, but...